Katie, but I'm here to introduce Dr. David Ray, who is a professor and associate chair within the Department of Biological Sciences at Texas Tech University. David is an evolutionary biologist who studies the impact of transposable elements on genome structure and function. He earned a BS in biology and master's degree in teaching for biology education from the University of South Carolina. After teaching high school biology, chemistry, and physics for several years, he returned to graduate school. David joined Lou Densmore's lab at Texas Tech in the late 1990s, where he began his work on crocodilians in collaborations with my advisor, Travis Glenn. David's fieldwork includes bats, crocodilians, and a variety of other amazing creatures. David's academic journey includes stops at LSU, West Virginia, and Mississippi State before returning to Texas Tech University, where in 2022 he won the President's Academic Achievement Award. This past summer, Dr. Ray was kind enough to work with me to annotate the tricky transposable elements in the tick genome I'm working on, and today he will be presenting transposable elements, a key to evolutionary biology and success. Please help me welcome Dr. David Ray.
I can't hear all the clapping. All right, so thank you, Katie. I appreciate that. Yeah, so I've been doing this for a while now, and it's right now I'm actually, so that you know, I'm in my backyard. We got a nice shed back here that we converted into a home office during the pandemic a couple days ago, and I have my dog next to me, which you'll see a picture of in just a few minutes. Because if I don't keep her in here, she'll be outside the walls barking, and it's a metal shed, so it reverberates if she's outside barking. So I know I've only got about 45 minutes here, so let's go ahead and get started. So yeah, I've been studying transposable elements for quite a while. I started studying them when I was a postdoc in Mark Batzer's lab at LSU, and it's been my whole career ever since. I like to joke with people that, you know, the people who study genes only get to look at about 3 to 5% of a typical mammalian genome. I get to examine about 50 to 70% of the typical mammalian genome. There's a lot more action going on with the TEs than there are with the genes in many cases. So I'm going to go through and talk a little bit just to introduce people to transposable elements if they don't. I'll shorten this down to TEs because it's just easy and that's what I'm used to saying. But transposable elements have been described by a friend of mine as the very unstable genius that genomes are made of. And in my opinion, he's absolutely correct here. They have a huge impact on genomes and genome evolution. And they come basically in two flavors. As you'll see on the left-hand side of the slide here, there are the class 1s, which are the retrotransposons, and the class 2s, which are the DNA transposons. And those are the two big major distinctions. And that major distinction mostly revolves around what their medium is through which they will replicate in the genome. So retrotransposons do exactly that. They retrotranspose. They make an RNA copy of themselves, and that RNA copy will then be reverse transcribed into DNA, and that new DNA copy will end up somewhere else in the genome. So by definition, they are copy and paste elements. They will always make a copy of themselves when they're replicating. DNA transposons don't always make a copy of themselves, but they often do. But they have a different medium through which they will replicate. They use a DNA intermediate rather than an RNA intermediate. And depending on the type of element that they are, they will have different kinds of DNA intermediates. And briefly, just a few different types that we may talk about here. There's ALU elements, which are the most common transposable element in human genomes and primate genomes. There are about 300 base pairs, and in our genomes, we have about 1 million copies of these ALU elements. So they make up about 10% of our genome. Line elements are also relatively common. They make up about 17% of our genome. There are a lot fewer of them, but they are much larger than the ALU elements. There's also a bunch of LTR retrotransposons and things like that. And then from the DNA or the class 2, there's things like hat elements and piggybacks and helitrons and helentrons and so on and so forth. And down here at the bottom, you can see the various relative sizes of these things. Signs like ALUs and BOVs tend to be pretty small, a few hundred base pairs, whereas lines and LTRs can be several thousand base pairs, and other ones are various sizes in between. And these are pretty standard characteristics by which we describe these elements. Now, as I mentioned, we have a million copies of ALU in our genome. So here, one of these is our human genome right here. About half is derived from transposable elements, mostly retrotransposons, but there are a few old DNA transposons in there. And this is relative, we're about mid-range for what a vertebrate might exhibit with regard to transposable elements. There's things like the lamprey, which has about the same amount. Fish tend to have generally fewer, but sometimes as much as half. Birds and turtles can have a lot less, but other organisms, have a lot more. It just depends on which organism you are. And again, I'll talk about that in more detail later on. And they also seem to be involved with genome size, which again, I'll get to in just a little bit. But this illustration doesn't really cover that particularly well. But they are major influences on genomic change, especially over evolutionary time. But you can also have a lot of change that occur, over relatively short periods of time. So one of those mechanisms of evolutionary change is just simply from the fact that they move, they mobilize, and they make new copies of themselves, and they move copies of themselves from place to place. So for example, here's the retrotransposons, that top part of the previous slide. And you can see that blue dot in the middle of that chromosome right there. And by definition, the retrotransposons will mobilize by making copies of themselves. And they'll move from chromosome to chromosome. They'll move from nearby. They'll move far away. It just depends, because what they do is they actually will make an RNA copy. That RNA copy is then exported out of the nucleus and then re-imported back into the nucleus and reverse transcribed back into the genome. So they can end up basically anywhere. Um, this this obligate copy and paste mechanism typically means that you will always get a new copy of these things. And if they are particularly active, that means your genome size will increase as a result of that. And this has been demonstrated over and over again through various studies, including one that I'm going to talk about in just a little while. DNA transposons, or class 2 elements, on the other hand, they can replicate and make a new copy of themselves when they mobilize, and some of them do obligately, but not all of them do. For example, in this case, the blue dot there is a DNA transposon. Maybe it's a hat element or a piggyback or a TC Marin or whatever. But typically what you're going to see is it's going to mobilize just by actually just physically moving from place to place. And occasionally it'll leave behind a copy, but that is dependent on when the mobilization occurs during the cell cycle. So it's not always the case that it will. So they are typically called cut and paste. And even when they do leave a copy behind, they're still cutting and pasting. It just has to do with when that mobilization occurs as to whether or not a copy is left behind, whether or not the replication fork has moved through that or not, that region or not. Rolling circle transposons like helitrons and helentrons and things like that, they are obligately copy and paste as they mobilize. And they're not as common, so I'm not going to spend a whole lot of time talking about them. Although we did find, Katie and I, quite a few of them in the dermacenter genome that we've been working with over the summer. So one of the things that we want to know about TEs is how often they, when were they deposited in the genome? Because that can be important as far as understanding how evolutionary changes occurred. And most of these, we can apply what's called the master gene model. And in the master gene model, what we get is we have a source element, in this case, a line retrotransposon that has its various components to it. It's got a 5' UTR, a couple of open reading frames, and so on and so forth. And at time 0, it's going to be producing copies of itself. It's a retrotransposon. It's going to deposit new copies throughout the genome. And at the time that that occurs, the daughter copies will be identical to the parent copies. And so at time 0, they should all look exactly the same. But obviously, over time, mutations are going to occur in all those copies, and they're not going to occur in the same way. So one element's going to get a mutation here, another one's going to get a mutation there, and so on and so forth. So that over time, what we end up is a bunch of different looking elements, but they haven't gone anywhere. They're still there in the genome. They've just decayed over time because of this mutational influence. And so we can thereby use that accumulation of mutations in the daughter elements over time, if we know what the original sequence was, which we can infer, and figure out how old these elements were when they inserted into the genome. And we generally do that using a plot that looks like this, a TE landscape. So this TE landscape, to read it, because you'll see some more of these, I believe, later on in a line format rather than just in the stacked bar plot here. What you'll find is that as we get closer to the y-axis, This is very, very recent. This is the example of time zero. Whereas if we go all the way back to here, this is time whatever, sometime far in the past, where these have accumulated tons and tons of mutations compared to the consensus sequence that originally gave rise to them. And I don't remember which bat this is. It's probably a Vespertilion, a Myotis, most likely. It's kind of sad that nowadays I don't recognize the bats by the picture of the bats, but I recognize the bats by the pictures of their transposable element landscapes. So that just tells you how many of these I've looked at over time. Um, so anyway, what you're seeing here is just an accumulation of various types of TEs over time. In this case, the purple ones represent signs. The lines are represented in dark blue here. The DNA transposons and helitrons are represented in orange and red, and then the green represents the LTR retrotransposons. So what you can see is in this particular bat, the LTR retrotransposons have pretty much ceased accumulating in this genome, whereas the signs seem to be pretty healthy. The DNA transposons are particularly healthy in this genome and are actively retrotransposing or transposing in the recent past. The second mechanism by which TEs can sort of change the genome is through actually causing genome rearrangements. And one of the main mechanisms is through ectopic recombination. As you can imagine, having 1 million copies of ALU through your genome is going to give plenty of opportunities for non-homologous recombination to occur, because the mechanisms through which non-homologous recombination occur, the enzymes involved recognize similar sequences of DNA. Well, here you've got 1 million copies of ALU scattered throughout the genome. That's a good substrate for causing recombination to occur. And so what happens during that process is there's a misalignment of the genome or the chromosomes as they line up during a synapsis, and you can end up getting a crossover occur. And as a result of that, you end up either losing or gaining bits of DNA as a result of that ectopic recombination directed by the transposable elements themselves. There are other ways that TEs can cause genomic rearrangements, but this is one of the ones that's easiest to explain. And then third is this idea of disrupting genes and regulatory pathways. So what they can do is if they land in the middle of a gene or land in the middle of a regulatory element, they're going to disrupt that gene or disrupt that regulatory element, or potentially even add a new function to that gene or new regulatory of TEs are the source of new regulatory elements. In fact, the mammalian placenta is actually thought to be the evolution of the mammalian placenta is actually thought to be as a result of a sign insertion that occurred long time ago at the base of the mammalian tree. So in this case, I'm not going to go through all these different mechanisms. You can read up on them as well as I can, but I am going to show you a picture of my dog. And this is her. She's laying right over there on her little pillow. But here she is out in the backyard chasing bubbles and all that kind of stuff with my daughter. But you'll notice her pattern here, the coat pattern. She's an Australian Shepherd, and she has what's called a merle coat pattern. And that merle coat pattern is actually derived from a transposable element insertion. In a gene called SILV, there is, in dogs, an insertion of a CAN sign in that gene. And depending on how long that insertion is, and that depends on the poly A tail associated with that sign insertion, you have different levels of this merle insertion pattern. And that's very common among dogs and also some other organisms. But so as you can see, there are phenotypes. There are actual physical phenotypes that derive from these insertions that occur throughout the genome. And that has led to these statements. One of them was from a paper that I was on many years ago. This idea that transposons and transposable elements are a major creative force in the evolution of mammalian gene regulation, as you can see with my dog right there. And then that leads us to this issue. The problem with transposable elements is that they're so ubiquitous. They're all over these genomes. And that makes it really difficult to deal with them. You're dealing with 3 million base pairs, all that look essentially the same. You got 1 million copies of ALU. I'm sorry, I said 3 million, 300 million base pairs of ALU alone in the genome. So when you're trying to map reads to a genome and 10% of them are coming from the same sequence over and over again, you could imagine the trouble that might be introduced as a result of trying to analyze those. And so for that reason, for many years, transposons were literally just masked out of genome assemblies as we got them. People just said, I can't deal with this right now. I'm going to get rid of them, and I'm just going to cover them up with ends in these genomes, and we're not going to deal with them right now. Now, fortunately, that is no longer the case. We have tools that can deal with that. Literally, one of the main tools that you find when you're doing a genome analysis is called repeat masker. That is the name of the tool, and everybody uses it. And its job was to get rid of these things so that nobody had to deal with them as a result. So, but they're important, and we don't want to do that. We don't want to get rid of them. And a couple of reasons for that are the hypotheses that I'm going to mention right here. One of those is this thing called the TE thrust hypothesis. So the TE thrust hypothesis is an idea that came about from Oliver and Green several years ago. It sort of gelled in the community for a relatively long period of time. But it's this idea that you have this set of sequences that are able to move from place to place. They take up large parts of your genome, and they can disrupt things in all those different ways that I talked about before. And that can act as an evolutionary force to increase diversity, genomic diversity, phenotypic diversity, all these different types of diversity as a result of that. And so if you look at the lower lower right-hand corner there, this is an idea that we were talking about a while back, and it's been around for a while, but there's this idea of fitness landscapes, right? So you have these various fitness peaks in your genome, right? So here's a combination of genes and a combination of genes, and that combination of genes right here gives this individuals who have that combination a certain peak fitness. And here's another peak over here and a certain other peak over here. And you can imagine doing single nucleotide substitutions to move from one peak to another is going to be pretty difficult because you got to travel all the way down that into that valley and then all the way back up again. But if you can initiate large scale changes as a result of transposable elements moving in a massive way throughout the genome, Matt Johnson, a colleague of mine here, suggested that that would serve as sort of a zip line from peak to peak as you go over evolutionary or over fitness peaks over evolutionary time. Ways of going from one peak to another relatively quickly without having to go into these valleys and traverse those and deal with all those selective pressures that are against that. The problem, of course, is that if you have too much transposable element accumulation, these are things that are disrupting genes. They are causing changes to the structure of the genome. So if you've got too much transposable element accumulation, you're going to have too much genomic instability, which can lead to a genomic meltdown. Whereas if you have too little, you've got too much genomic stability, which means that basically the genome is in stasis and you can only rely on small changes in order for evolutionary change to occur. Whereas if you've got this Goldilocks zone here, this region where there's some transposable element activity, but not enough to kill you, and not too little, then you can have this area of genomic dynamism, where you can have transposable elements thrusting organisms into new niches, causing adaptive change that they can take advantage of. And I'll give some examples of those a little bit later as well. Now, there's other hypotheses as well. Another one that's really popular is the epitransposon hypothesis. This is the idea that we don't see transposable elements mobilizing a whole lot in most of our cells. In somatic cells, they're pretty much epigenetically silenced. They're methylated, and that methylation shuts them down and prevents them from moving from place to place. And that's a good thing, because if that doesn't happen, then you tend to get cancer. There have been several examples of transposable elements that have managed to mobilize in, say, a lung cell, and that lung cell, because of that mobilization, it shuts down a tumor suppressor gene, and that tumor suppressor gene, with it being shut down, the cell basically turns into a tumor. And you can actually look at the tumorous tissue and the neighboring healthy tissue and identify the transposable element insertion that may have caused that tumor to occur. And in fact, if you look at the top of my head, just 2 weeks ago, I had a carcinoma removed from the top of my head. It was really gross, but it was fun because they let me go into the histology lab and see the tumorous tissue. It was actually, for what it was, I was actually having a good time. But anyway, so I asked them, I was like, can I have some of that? Because I want to sequence that tissue and figure out if it was some sort of change. What was the change that caused that carcinoma to occur? Unfortunately, the person who could grant me permission for that wasn't there that day. So, if they have to come back and get some more, I'll ask again. But anyway, this idea that TEs are epigenetically silenced is important because we don't want them mobilizing just crazy in our genome. The problem is that under stressful conditions, epigenetic changes can be changed, they can be modified. And so in some cases, what you find is that as stress occurs on an organism, you basically release the kraken of transposable elements by undermethylating those regions, allowing those transposable elements to start mobilizing, thereby inducing potentially beneficial change, but also potentially bad change as well. But obviously, the bad change will be selected against, and the beneficial changes will be selected for. So those are the ones we'll get to see in the future. So these are important. We need to know what they are. We need to know where they are. And so that leads to this idea of curating transposable elements. This is what Katie and I have been doing all summer, is curating and figuring out what transposable elements are there, what are they doing, how are they distributed, et cetera. And it's really, really important that we do that because we got to know what's going on. What was this change induced by a transposable element, or was it not? And what kind was it, and what kind of things can we figure out from that? So several years ago, almost 10 now, 8 years ago, we wrote this paper called, Transposable Element Curation is Vital to New Genome Assemblies. I can't remember the actual title. But the premise behind that paper was people are doing this wrong. People are not showing enough care to accurately curate the transposable elements in a genome. The typical tool is that if somebody generates a new genome assembly, they'll run a program called Repeat Modeler, and then they'll take the output of the Repeat Modeler and throw it into Repeat Masker and analyze the genome, and that's the end of the process. And in a testy email to some colleagues, I quote myself here, everyone should stop throwing raw Repeat Modeler output at newly sequenced genomes. They are bad people if they do so. And I stand by that. People should pay more attention to these things and not just do that. And I'll show you why in just a second. Here's why. It's because you're going to be missing things. You're going to be inaccurately reporting things. You're not going to be doing a good job identifying these things. So because as much as I love using Repeat Modeler, and I'm good friends with the people who wrote it. In fact, I was exchanging emails with Robert and Arian just this morning. Just for example, in Noctilio leprinus, the bat that you see right there, 15% of the IDs from Repeat Modeler are wrong. They're just incorrect. And in Crazy Nictus thonglongii, almost 20% are incorrect. And if you look right down here, let's say this little, it's hard to see, but this little colored line right down here, that represents the output from Repeat Modeler for some transposable element, some presumed transposable element, right? But then when you actually go into the genome and look at it, what you find is that consensus sequence from Repeat Modeler was quite incomplete. The actual consensus sequence that this is a representative of is over 200 base pairs longer. And if you can imagine, if you're missing 200 base pairs of that element, and there's 1 million copies of that element in the genome, you're missing 200 million base pairs of annotation in that genome. It's going to be inaccurately annotated as a result of that. If you've got a 2 gigabase genome, that's 10% of your genome that you're just simply getting wrong. And so it's really, really important that we accurately curate these TEs in these genomes. And Robert was a little bit more measured in his email. He says, yeah, the bottom line is that Repeat Modeler took us from doing these things in a year to a couple months, but still, you need to do a better job than that. And here's a good example. Oh, I forgot about this slide. Also, doing it that way is a problem because you're using homology as an annotation mechanism. And so imagine, if you will, you've got two genomes. They both have some element in it. Let's call it a line. Genome 1 has its line that's colored in blue. Genome 2 has its line that's colored in green. But the problem is that if you use this element that we know about in genome 1 and try to annotate genome 2, what you're going to find is that they have evolved in different paths after the common ancestor. And so they've accumulated their own set of characteristics over time. So if you try to characterize genome 2 using elements from genome 1, you're going to get an inaccurate picture. In other words, you're going to see the ones in genome 2 as being older than they actually are, because we have to deal with the fact that that divergence occurred some time ago. And so that gets worse as you get older and older and farther down in the phylogeny. So it's really important to annotate these things properly. The best example I have, there's the title of that paper I was talking about. Best example I have was from this butterfly, Heliconius melpomene. And Heliconius melpomene, this is what you get for a TE landscape if you throw raw repeat modeler out at this thing, right? You see it's got a weird shape to it, first of all. But you see there's a bunch of lines, but they're all old, and there's a bunch of DNA transposons. Some of them look pretty new. There's basically no signs in the genome as far as we can tell. And LTR content, nothing really to speak of. But then we did a good transposable element curation of this genome, and this is what we see when we're done. Um, First of all, we missed an entire family of sign elements that nobody knew about before. It's there. It's quite obviously there. That was just completely missed if you throw raw repeat modeler output at this thing. So in addition to that, we see a different pattern of the DNA transposons. Instead of having this old accumulation here and some new accumulation, we see a lot more new accumulation. We're pretty spot on with regard to the LTRs, but the lines, not so much. There's not a whole lot of line accumulation occurring in this genome compared to what we thought we saw with raw repeat modeler output. And so it's important that you do this in order to get this. So that was why we were asked by the Broad Institute to come on board for what's called the Zoonomia project. The Zoonomia project finished a year or two ago, and our job was to take the 248 mammal genomes that they were generating through their project and do the de novo TE curation on them. The person who led this paper is Austin here, picking up a snake of some sort. I'm not sure. I can't tell what it is from here. But anyway, this whole project was led by Sherston and Eleanor. And fortunately, we were able to get a really nice science paper out of this last year. So our job was to curate 248 mammal genomes representing 109 mammalian families. And it took a long time. It took us 2 years. And there were a total of 9 of us in my lab working on it, plus some colleagues outside. And we spent a whole lot of time looking at screens and curating these TEs manually. It took a very long time, between 1 and 19 rounds of curation, depending on what species that person happened to be working on. And we ended up, though, after doing this and after corresponding with several others, with 8,000 novel consensus sequences that nobody had ever seen before in mammals. Most of them were LTR, some of them were lines, some of them were DNA transposons, and so on and so forth. But there were a buttload of them, and we were able to get that done. And again, we got rewarded, with a couple nice science papers out of this. So here was one of the major figures to come out of this paper. What time is it? This was referred to, I have to say this was referred to by reviewer 3 as a tour de force, and I'll never stop telling people that, because you very rarely get a nice set of words like that coming out of reviewer 3. But anyway, so to look at this figure, it's a busy one, but it's an important one. What you're seeing here is the mammalian tree that came out of this effort from Zoonomia. And here are the major clades. Here's the rodents and the primates and the apotherians, and there's the cetaceans, and here's carnivores, etc. But at the tips of those what you're going to find is these little whisker patterns here. I don't even know what to call this, the ends of the tree. But what you're going to find is that there's going to be a dot there that tells you what type of transposable element was the one that was most prevalent in that genome. What you're going to see next is the total TE proportion, how much of the genome is actually represented by transposable elements overall. But then we were also interested in what has been happening more recently. What has been happening in these different clades over the more recent past? And so that's represented here by the final tip on this thing. So we've got dominant TE, total TE content, and finally more recent TE content in that genome. And so we focused on that recent TE content. We can't, estimate the mutation rate for every single organism and plot that onto this giant tree. So instead, we used the average mammalian neutral mutation rate, which is 2.2 times 10 to the negative 9 substitutions per base pair per year. On average across the mammals. And that, by focusing on things that were only 4.4% divergence from their consensus, allowed us to focus on things that were probably around 20 million years old or less, right? And so that's what this up here is representing. All right, so Obviously, with a big project like this, we have to make sure we're doing it right. And so we have to focus on quality control. We got to make sure that we're curating these things accurately. So, did we miss anything due to that recent accumulation cutoff? So, to test for that, we wanted to make sure that we were seeing things we should be seeing. For example, there are lots of different TEs that have been examined before. They are common to all mammals. There are some that we knew were common to this group or that group. And we did indeed find all the things we expected to find. We also found a bunch of new stuff, but we found the things we expected to find. So that's good. That means we were on the right track. All right. Um, we also wanted to see if we were finding things that we shouldn't have found. Are we finding things that, don't necessarily represent real transposable elements? And that's a little bit harder to do. So what we decided to do was take advantage of the fact that in some organisms, we know that there are certain TEs that are exclusive to that organism. In this case, we're going to deal with helitrons. Helitrons, among mammals, they are exclusive to one family of bats. That's it. We don't find them in any other mammals as far as we know, and we continue to say that now that we've actually finished this project. They're found in this Vespertilianid bats, and that's it. So we thought, okay, if we're finding helitrons elsewhere in this tree, they're either real or not real. We'll take take a look at them and see if they actually are real. But it turns out they weren't real. This is actually exclusive to Vespertilianid bats, this one family or this one superfamily of TEs. So we found a very, very low occurrence of them outside of Vespertilianids. And whenever we looked at those occurrences outside of Vespertilianids, once we looked at the actual sequence and compared it to consensus sequences, it was obvious this was just a false positive of some sort. So in addition to that, we wanted to make sure that the actual assembly quality wasn't influencing whether or not we were accurately getting the TE consensus sequences. It turns out it's fine. There was no difference in our ability to detect TEs from either the quality based on contiguity of the genome and 50 or the completeness of the genome with BUSCO. So we weren't worried about that. So it turns out that our methods were pretty good. And some of the takeaways from this work are that genome size, first of all, does continue to correlate with the accumulation of transposable elements. There's a general trend that as you have more transposable elements, not surprisingly, the bigger your genome is. It's not super strong, but it's there, and it's something that's consistently found. So last time I gave this portion of this talk, I was in Ireland at an actual conference. And you know at conferences, they give out drink tickets. So I decided to make this part into a game show. I wish I could do that with you guys. If I was there in person, I would. I would, have some candy or something I'd throw out. And I did this as sort of a game show part of the talk, where I'd say, okay, who do you think is the winner or the loser in this particular category of TEs? And I don't drink, so I was quite happy to give away my drink tickets. So we can't do that here, but I'm going to, go through this anyway. So the winners and losers among mammals, who has the most TEs, who has the least TEs, who has the most of this particular type of TE, and so on and so forth. So the big winner, actually the big loser among all mammals is the star-nosed mole. It has very little transposable element content compared to other mammals. It's only about 27.3% transposable element. The genome only has about 27% transposable element content in it. So it doesn't have a whole lot of its genome taken up by transposable elements. Now, within subcategories of that, for example, line signs, LTRs, DNAs, and rolling circles, we have our losers as well. All right. One of them is the Gambian pouched rat. It has the lowest line content of any of the mammals, except of course for the potentially for the star-nosed mole. The loser with regard to signs is the Malayan pangolin, has the lowest amount of signs among all the mammals. LTRs, the Indo-Chinese shrew. The European hedgehog has the lowest DNA content, and the guinea pig has the lowest helitron content of any of the mammals that we were able to look at. So now those are the losers. Let's go to the winners. What organism has the most transposable elements in it? It turns out it's the aardvark. Uh, it's funny to say, and it's a cute picture as well. So the aardvark has just a gigantic amount of transposable elements in it. I can't remember the actual total amount, but it's something around 70%. And most of that is lines. So the line content is around 52% in that genome. The biggest sign content among mammals is the hazel dormouse. When I gave this talk in Ireland, that actually is a native organism to Ireland, so I was able to wow the crowd with my knowledge of their local fauna. Um, the northern mole vole, which is always just fun to say, has the most LTR retrotransposons. And not surprisingly, a couple of bats, at least not surprisingly to us, a couple of bats are the ones that have the most of the class 2 elements, DNA transposons and rolling circle transposons as well. The bumblebee bat being the smallest mammal that we know of, but yet has a huge amount of DNA transposons in their genome. And not surprisingly, the tube-nosed bat for us is a vespertilionic bat because it has those rolling circles in it. Um, so we decided we were going to look at this from a community ecology perspective and say, okay, if we assume that transposable elements, because they can mobilize, they can essentially reproduce inside a genome, they can act as organisms that live inside this ecosystem that is a genome. And we decided we were going to measure how much TE diversity and how much TE content using these ecological tools, things like Shannon diversity and Pylo's J. I hope I'm pronouncing that right. I've never been able to figure it out. But Shannon diversity is a way of measuring diversity of organisms, but we applied that to these transposable elements. Pilou is also a measure of diversity we applied to transposable elements. The difference between the two is relatively subtle, but Shannon diversity tends to give a lot more weight to the most commonly occurring species in an ecosystem, and also in this case, the transposable elements, whereas Pilou we wanted to include because there's a difference, right? You can have two organisms that have the same amount of TE. Both organisms have 50% of their genome is TE derived. But if one of them has its 50% from one type of element, that's different from having an organism whose 50% is derived from 8 different types of elements. That's a different kind of diversity in those ecosystems, those genomic ecosystems. So we wanted to do both sorts of measures. And that also ended up on a tree, another tree that took forever for Austin and Matt to get put together. I'm not going to spend a whole lot of time on this slide because it's just a bunch of information. But again, if you look at another figure from that paper, what you'll find is there's various rings in this tree. And as you work your way out, you still have the dominant TE type, the recent TE proportion, and the diversity, both in Shannon's and Pylo's indices, for that diversity as you go. And some of the things that we found were that if we look at these from a community ecology perspective, we find that we can get these plots here. And these plots tell us, based on the proportion of recently accumulated transposable elements, how much diversity do we see with regard to the TEs in those genomes? And generally what you see, especially with Pylos, is an inverse relationship. What you find is that as TE accumulation goes up, the amount of TE diversity goes down. And what that means in simple terms is that as you get more TEs, it tends to be one type of TE that tends to dominate. So one, typically, if in an ecological perspective, there is one dominant species in a typical genome with regard to transposable elements, a line or a DNA transposon or one type of sign. That one tends to dominate all the others and essentially overwhelm them with regard to the signal that we see for these diversity indices. And that prediction works really well generally for mammals. As you have more transposable elements, you tend to have more of one particular type of transposable element rather than a bunch of different ones. It doesn't work so well with those bats that I talked about, those vespertilionid bats. Those vespertilionid bats and some other bats have a lot of DNA transposons. That is completely unexpected. In fact, when we first discovered these things way back in 2005, 2006, I was working with these bat genomes, for whatever reason, and I started finding all these DNA transposons in them. And at that point, nobody expected them to be there because every other mammal we had looked at did not have any active DNA transposons in their genome. And all of a sudden, I'm finding these bats that have all these DNA transposons that seem to be bouncing around all over the place. I sat on that paper for several months because I was convinced I was wrong, and then finally sent it in. And it turns out we beat Cedric to publication by about a month. as far as finding exactly the same thing for this. So it was real, and but I was really careful. I was very nervous about that paper. Um, so we do have this case of DNA transposons occurring in some groups of mammals, in particular bats. And there are other groups that have DNA transposons that have invaded them. In particular, we've got some cetaceans that have them. Some of the primates have them. There's the star-nosed mole, and there's Echinops, which is the tenrec. And they have some DNA transposons, but nothing like what we see in the bats. The bats have just a huge amount of DNA transposons that the other guys don't have. They're there, don't get me wrong, but the bats seem to be exceptional. But we can't just say seem to be exceptional. We need to actually investigate this. And it turns out that this is actually real. So this is coming from that same paper. I'm going to talk about it a little bit more. But it turns out that this ability to take on DNA transposons seems to be associated with diet, in particular insectivory and carnivory. So we did an analysis here to investigate the distribution of DNA transposons in the various taxa. And what we find is that, sure enough, if you are an insectivore or a carnivore, you tend to have a higher capacity for taking on DNA transposons than other mammals do, than the herbivores and the excuse me, herbivores and maybe nectarivores or whatever, frugivores. And that probably seems to be associated with viruses, which will tend to move things from genome to genome. It probably has to do with predator-prey interactions. You're encountering your predator or your prey's body fluids more often, allowing for the exchange of genetic information potentially as you go. The cellular machinery is similar among organisms that are predator and prey because they're both mammals, whereas if you're eating plants, that's a very different set of cellular machinery, which means you're going to have a different set of transposons, which are not going to necessarily want to move to an animal genome. And it also may have to do with the cellular defenses against transposable elements, which we just don't have time to get into in this talk today. All right. So real briefly, I want to go through just one or two more stories about the way that this works. So like I said, we can't just say that chiropterans, bats have more DNA transposons. We need to actually investigate and figure out how that happens. So this is Nikki. She's another graduate student. She should be finishing up this year. And she wanted to know how many horizontal transfer events have occurred for DNA transposons in bats compared to other mammals, and what other organisms are involved in those horizontal transfers. And so this has occurred to other people before. We actually published on this many years ago. Another group had published on this with fish many years ago or several years ago. But we wanted to focus on the bats and figure out how did these MFs get here, right? That was a quote from Nikki that I wanted to include on this in this slide. And so what she did a bunch of bioinformatic processes in order to investigate this. I'm not going to go through that because I'm running out of time. But what we found is, yeah, sure enough, the bats do appear to be exceptional. They do appear to have a very high capacity for taking on DNA transposons through horizontal transfer than other organisms, in particular other mammals, do. It's substantial, 229 versus the next highest, which was only 15 horizontal transfer events that we were able to identify. So she built this tree for those 229 putative horizontal transfer events, and we found a few things that most of them were DNA transposons, sure enough. Most of them, over half of them, were in Vespertilionids. I keep on bringing this group up because they're pretty important in bats. There were 8 that transferred into some other clades, Rhinoliphodia, and so on, in Vespertilioidea. And most of them, we don't know where they came from. They appear all of a sudden in this group of bats or this species of bat, and we have no idea where they came from. For the ones that we are able to determine where they came from or they were exchanged with, we don't know the directionality. They came from Afrosaurids, tenrecs, lemurs, tree shrews, some lizards. But one of the most interesting ones, in our opinion, is this one from a fish. Somehow, a transposable element was able to horizontally transfer to or from Pipistrellus cooli, which is a European bat, and an African reedfish. And this is the alignment of those sequences. It's a 2,291 base pair consensus sequence. And between these two organisms, there are only 8 nucleotide differences. Only 8. So this isn't a vertical transfer. This isn't something that was shared in all of the ancestors of these guys. Somehow that element managed to move either from the bat to the fish or from the fish to the bat through some sort of an intermediate. And we could show that by comparing to the sister species of Pipistrellus cooli, Pipistrellus pipistrellus, which shares much of its habitat. And those two organisms they diverged from one another about 15 million years ago, but yet there are no instances of this element in Pipistrellus pipistrellus, and there are 347 instances of this element in Pipistrellus cooli and 52 elements in Herpetoichthyes, the reedfish, but none here. So that tells us that somehow that element was transferred between these two organisms. How has been the subject of discussion for our lab for quite a while now. You got ideas, let me know. Most likely viruses or some sort of predator-prey interaction, but somehow that physical distance had to be traversed as well. So we don't know how that happened. Hopefully, by the time we get every organism sequenced, we can actually track it down. All right. So I mentioned the TE thrust hypothesis a little bit ago. One of the predictions of the TE thrust hypothesis is that if you have recent TE accumulation, you should see recent species diversification as well. And we see tantalizing hints of this, that as transposable elements start expanding through your genome and start accumulating in your genome, that's going to cause all the disruptions and potentially lead to adaptive change. So what we should see is recent species diversification occurring alongside that. And we kind of do see that. There's tantalizing hits, right? For example, there's this genus Rosetus, which only has 7 species, and it's pretty much a flat line with regard to transposable elements over the past 50 million years. Similarly for Corolia, only about 10 species, flatline, right? Whereas Hipposideros and Myotis are 2 very species-rich groups of bats, and we see lots and lots of transposable element accumulation. The problem is that when we actually measure this using statistics, we don't find too much that's actually statistically significant. We do find that there does seem to be an association with LTR signs and rolling circles, but not so much lines and these. And it's really not all that strong. So what we're hoping to do next is actually take advantage of a project that I'm involved with, which is BAT1K. And our objective with BAT1K is to sequence and assemble the genome of every living bat species over the next 15, 20 years or so. And we've just made a good start, right? So here are the various phases that we're planning on going through. We finished the pilot study a couple of years ago, and literally 2 weeks ago, we actually finished phase 1, which is to sequence and assemble the genomes of 103 bat species, each one, or at least one representative from every bat family. If you're interested in participating in this, the data are there and available. You just have to make sure that you agree to the rules for sharing. But it's there, and we'll happily share it with you as long as you agree to those rules. Let's see, do I have time to go through this? Katie, tell me, do I have time to go through this? It's only like 3 minutes.
Do it.
Okay, do it. All right. So one last project before I wrap this up. This is part of an award-winning presentation from Francisco there. He was literally in Guadalajara last week, and I'm going to give a portion of his slides here that he won $500 for at that presentation. I had him given to my class when he got back. But what Francisco's working on is using transposable elements as phylogenetic markers. This is something I worked to develop many, many, many years ago when I was still a postdoc, taking off from work from Norihiro Okada, from even before that. But the idea is that we've got all these TEs in the genome. Can we use them to infer phylogenies? Because that's kind of important. We're evolutionary biologists after all, and we need to know how organisms are related to one another in order to interpret evolutionary change. And Francisco has sort of taken that idea that I was working with as a postdoc and started, is expanding it given the new environment we have with all this new genomic data. So just to give you a little bit of background, because transposable elements accumulate over time, we can use them as phylogenetic markers by identifying insertions that might be shared among all 3 species in this case, or identifying insertions that are lineage specific, or identifying insertions that may be shared between these 2, but not that one right there. So it's a typical phylogenetic marker that you might imagine. And it has an advantage in the fact that they don't get removed from the genome. And we know what the ancestral state is. We know the ancestral state is the absence of the element. And so since they don't get removed typically, and we know what the ancestral state is, they make the interpretation of the data much easier. But what we want to do is, I was doing that as a postdoc, looking and identifying these things either through hybridization or through some simple bioinformatics. But then we would actually do PCR. But now there are hundreds, if not thousands, of genome assemblies out there that we can directly compare to one another. And we want to take advantage of that rich data source in order to figure this out. So we can identify in each of these genome assemblies all the signs that exist in those genomes using repeat masker. And then we can align those genomes to one another using something like progressive cactus or some other things. And then we can score all those signs as to their presence or absence in each of these genome assemblies. And we end up getting a matrix that looks, like any other phylogenetic vectors that you might imagine. And so what Francisco did over the past couple of years is he built this whole genome alignment for 28 bat species. It involves 1.3 million sign insertions. That's just the signs, not the lines, not the LTRs, not the DNA transposon, just the signs. And there's 1.3 million of them that he had to deal with. And that alignment itself took over 2 months to build using progressive cactus. So he was getting to the end of his master's thesis. He didn't want to stay in his master's degree for another 3 years to analyze all 1.3 million of those. So instead, what we did, we decided to do a 4-genus proof of concept. Does this actually work? And we picked these 4 species right here: Molossus molossus, Eptescus fuscus, Antrozous pallidus, and Myotis myotis. These are 4 species that we have a pretty good idea what their phylogenetic relationships are. Can we get that same relationship back by looking at the signs? And so he built his matrix, as we talked about, and looking at that matrix, using these 4 species, we get a bunch of different potential phylogenetic hypotheses. And here's one of them. The one that's most prevalently represented by those signs, down there at the bottom, you see there were 7,300 instances that supported this phylogenetic relationship, with Molossus being the outgroup, Myotis and Eptescus and Antrozous having that relationship there. But that wasn't all we saw, right? We also saw this one, which suggests that Eptescus and Antrozous are closely related to one another, but it doesn't tell us anything about Myotis and Molossus. Similarly here, this tells us that Eptescus and Myotis are actually more closely related to one another, but Molossus and Antrozous, we don't know. And so on and so forth, through all these different characteristics, or all these different columns in this matrix. And so using that information, we could say, okay, it looks like, according to this data, that this tree right here is the most likely phylogeny. But we do see substantial numbers of insertions that are giving us a different picture. And what that suggests is that there was some sort of ancient introgression or relatively modern introgression occurring in the ancestors of each of these groups. And this is a way of detecting that sort of thing in the genomic data that we're seeing. So I'm excited about this. Right now, what he's doing is this involves several tens of thousands of transposable element insertions, several tens of thousands of TE sign insertions in these 4 species. Imagine trying to do that with that 1.3 million, right? And even then, what we did for this was we made a major assumption that our alignment was right and that we were calling these things. We just did a very simple way of calling these things, and we assumed that we were right about that. The problem is that that's a major assumption. Maybe maybe our methodology was wrong. We need to test that and figure it out. So what Francisco is working on right now is actually trying to incorporate machine learning to identify what the characteristics of a given sign insertion are. What are the characteristics of it being present in one species but not present in the other species? How can we interpret that whole genome alignment so that a machine learning algorithm can go in and identify these things with relatively high accuracy? It turns out that we had relatively high accuracy, because after doing this, Francisco went in and selected about 1,000 of these things and actually physically looked at all of them with his eyes. And we were accurate to about 90%, right? So that's good. It means our phylogeny is probably right. But by incorporating machine learning, what we hope to do is improve that even more and have the machines tell us exactly which ones are insertions and deletions. So I'm going to wrap it up there. This is I have three students who actually live outside the state, in Texas, well, outside of Lubbock, in Texas, in Florida, and in Alabama. So all of our lab meetings are in the Zoom. So we decided to take this picture recently of our Zoom meeting for our Monday lab meeting. And you've seen some of these people here. They're great people, and I love working with them. And hopefully we can continue that for at least until they finish their degrees. But for now, I have to ask you, do you have any questions? How'd I do on time? Oh,
You're doing okay. We'll do about 5 minutes for
okay.
questions, but thank you, David, so much. That was a really great presentation.
You're welcome.
I
Anne has
learned a
her
lot.
hand up.
I'll give time for about 3 questions so we can end at 12:30. It looks like Ann Summers has a question. Go ahead, Ann.
Yes, I do. The alleged virus origin, the host of SARS-CoV-2 in
Mhm.
China. Uh, do you know what family of bats that was?
That it was originally identified in Rhinolophus, that genus. I can't remember what species, maybe Hippus, Rhinolophus sphaeromequinum. I don't know, but it was originally found in, I believe, a Rhinolophid species. I don't know what species is. I could probably look it up and find that out.
Well, I was
Uh,
just wondering if it's possible that things like long COVID could be a disturbance of our own or mammals, another mammal's transposable elements.
I suppose that's at least a hypothesis worth looking at, but I'm not a medical person, so I have no idea about the answer to that. I had
And
COVID. I had it really bad. I was in bed for about a month. So I don't want to get it again. And I'm doing everything I can to avoid that.
listening to you, the words Marburg, Ebola,
Uh,
all
yeah.
that other kind of stuff was running through my head. What a brave fellow you are.
Well, yeah, I did. I have gotten sick a couple times. I was crawling in the floor of a cave up here in Shamrock, Texas, trying to collect some myotis. And about 2 weeks later, I ended up again in bed. And I think I had histoplasmosis. I'm not sure. But yeah, but it's just too much fun. Ask Travis. He likes catching stuff and getting bit by things. Um,
Yep. I got a bunch more questions. I'll email you.
okay, thank you.
You and we'll do 2 more questions. It looks like there's one in the conference room.
Oh, hello. I'm Olivia in the conference room. Thanks for your really good talk. I was wondering, I know I study fungi, and the one thing I hear about bats is that they get a lot of fungal infections. And I also know that, some fungi have a lot of transposons as well. So I was kind of wondering if uh, you're looking at all at like fungal transposable elements and if they're similar to like the ones that are infecting bats and if they're similar to these ones that you're seeing sort of these unknown mysterious bat transposons.
Yeah. No, we haven't looked at that. If I could, if I had, 24/7 a day to just do what I do, which is look at all these TEs. I had the greatest summer this past summer because we had that data freeze from Bat1K of 103 assemblies and I took the summer to not do any teaching, to not do a whole lot of traveling, and I just sat here in my office and I looked at transposable elements all day long every day, and it was the best summer I've had in a long time. So if I could do that with fungal genomes, and right now we're doing it with tick genomes and all that kind of stuff, I would. I would not be surprised if we saw something like that. But who has the time? That would be great. Thank you, Olivia. Did I answer your question?
Yes.
All right, we'll take one last question.
I have a quick comment.
Sure.
Yeah, go ahead.
Yeah, we do this kind of stuff with bacteria,
Mhm.
and starting out with a single cell, a single colony, and sequencing it. And things are jumping while they're growing up for you to isolate the DNA. I mean, it's happening continuously
Yeah.
in at least the Enterobacteriaceae that we see. So that's a really your elimination of the transposon thing is really if people are using that, they're really missing a whole lot of stuff in bacterial genomics as well.
Oh yeah,
Having
definitely.
this little conversation with the people at NLM. So
All right, looks like we need to go ahead and close up since the next class is trying to get into the seminar room. Thank you again so much, David,
No
for
problem.
presenting today, and happy weekend, everyone.
All right, everybody have a good day.
Thanks
See
a
ya.
lot, guys.
Thanks, Katie. Hope she's gone. And if you have other questions, let me know. I'll be happy to talk to you by email.
Oh, yeah, your email address must be there somewhere. So
I'm sure it is.
yeah, I'll send you a copy of my journal club that's planned for the spring. So
All right. Have a good day.
yeah, bye-bye.
Bye.